Okay. Did everyone sign in? I think everyone signed in. So I'll just move on to, okay, Unit 4. We're getting through it. Circulation and blood pressure. Same as every week, my office hours are 2:30 to 3:30. I haven't seen any of you there, so if you want to come, have some questions, I can help you out. Also email me if you have any questions, and my notes are on Husky CT. And if I forget to post a recording, email me because I think I forgot to last time, and I'll put it up. But all of the recordings are up now, and I'll post this one. Okay, so starting off with an overview of the vessels. So this is just like the path of blood from the heart to the tissues and then back to the heart. So as we kind of went over in the last few units, blood is oxygenated in the lungs, passed back to the heart, and then is sent out to the body through first arteries, and then the arteries get smaller into arterioles, and then the arterioles get even smaller into capillaries on like the surface of tissues. And that's where gas exchange occurs. So oxygen will come out of the blood into the tissues, and carbon dioxide from the tissues will enter into the blood. And now blood is deoxygenated, is carried back up through venules, which are just smaller veins, and then those merge into veins, and then we're back at the heart again. So here's some review from last week, just to help you remember. Arteries always carry blood away from the heart, so arteries away, and they usually contain oxygenated blood. Veins always carry blood towards the heart, and these usually contain deoxygenated blood. Does anyone remember the exception that we went over? I think it was last week.
Pulmonary arteries.
Yeah. So the exception is the pulmonary. So the artery still carries blood away, and the vein still carries blood towards the heart, but it's the opposite with oxygenation. So I was gonna do like a drawing, but I drew it on my iPad just to like emphasize like artery to arteriole to capillaries to venule to vein. And then this diagram also shows you the difference in the thickness of the vessels. So the arteries are really thick and have a smaller lumen, which is just like the inside of the vessel, where the veins have a thinner wall and are more oval shaped. So that's just a visual of just general circulation. Okay, so now I'm gonna go into each vessel individually. So first we have arteries, and then this is some general features. They have a thick tunica media, which is the middle layer of the artery wall. And this is made of smooth muscle and elastin protein. So the fact that it has a lot of smooth muscle in the wall makes it easy for the body to regulate the thickness, the lumen, so the radius of the vessel. Since there's smooth muscle, the body can then either constrict or dilate the arteries to regulate blood pressure overall in the body. So there's a round lumen or space in the middle of the artery, and they can retain their shape in the absence of blood flow, which also helps to maintain overall blood pressure. Another important feature is that they have a lower compliance, which basically means that they're less stretchy. They're more stiff. So there are two types of arteries. Elastic arteries have in even lower compliance, so they're more rigid. So these ones stretch and recoil really quickly because they're stiff. So they help to create like a pumping action. An example of this is the aorta. So when blood fills the aorta, it like recoils really quick, which helps send the blood out through the body. So in other words, it acts as a conductor of pressure. So it creates a pressure wave that helps to send blood throughout the system because once blood fills them, they recoil really fast, which helps blood be like propelled forward. The second type of artery is muscular arteries. These ones have a higher compliance, still an overall low compliance though because they're an artery, but higher than the elastic arteries. So they're a little bit less rigid. So there's less resistance to blood flow in the muscular arteries. Moving on to arterioles. Arterioles are just smaller arteries that our arteries branch off into. So these are really important for regulating overall blood pressure and for controlling perfusion of the tissues. And perfusion just means to supply blood to. So a very important feature of arterioles is that they regulate total peripheral resistance, or TPR. And this is the resistance created by the arterioles, which is determined by their radius. So arterioles, we'll see, are regulated intrinsically and extrinsically, which then changes the diameter of their radius to either allow more blood flow to certain tissues or to restrict blood flow to certain tissues. That's. Does anyone have any questions at this point? Okay, so I just wanted to think about this a little bit more deeply to see if we can reason through it. So during the fight or flight response, the body may dilate arterioles around organs like, what do you guys think? During the fight or flight response, where might the body want to send more blood?
Towards the heart, and the brain.
Yeah, that works. I think I had another one. Yeah.
Maybe the lungs.
Yeah, lungs works. I think I might have also said skeletal muscle. Yeah, 'cause during the fight or flight response, like you're in survival mode, you got to get more blood and oxygen to those vital organs like the brain, heart, lungs, skeletal muscle, so you can like move and get out of the situation. The body, during the same response, may constrict arterioles around what type of organ? Anyone have any ideas? Yeah.
Like the GI organs.
Yeah, exactly. It's like we don't need to focus on digestion right now. We're trying to like get out of a bad situation or something. So on the other hand, during rest and digest, the body may dilate arterioles around what organs to increase blood flow to this region? We kind of just said it, so I'm just gonna put GI organs, because rest and digest, more blood flow to the intestinal tract to like help digest food. Yeah. And then this just relates to what we just talked about. So actually altering the radius of arterioles to control blood flow is referred to as the total peripheral resistance. And that's like what TPR is. So, like I mentioned before, the body regulates the arterial radius to alter blood flow to certain regions. So it does this intrinsically and extrinsically. Intrinsic regulation is at the more local level. So intrinsic regulation refers to mechanisms from the tissues themselves. So blood flow is adjusted just to meet the needs of some tissue that might need more blood perfusion to it. So it's a local effect, and it does not affect the overall blood pressure in the body. So two things that could happen to the arteriole during intrinsic regulation are vasoconstriction or vasodilation. Vasoconstriction is contraction of the smooth muscle, so that reduces the radius of the arteriole, and that happens when the tissue needs less blood. So the arteriole will constrict, less blood will flow into the capillaries, and vasoconstriction is initiated by high oxygen levels, low carbon dioxide, and a high pH in that local tissue. So if we think about this for a second, if a tissue has a lot of oxygen, it doesn't need more blood flow to bring more oxygen. So we can constrict and relax and send the blood somewhere else. Low carbon dioxide means that we don't need a lot of blood to come and pick up the carbon dioxide. So that's the reasoning behind vasoconstriction. Vasodilation is just like the opposite of that. So it's relaxation, increasing the radius, and increasing blood flow. So this is done when we have low oxygen levels, because we need more oxygen in the tissue, so we want to increase the radius to allow more blood to the region. High carbon dioxide and low pH. So then to regulate kind of overall blood pressure, we have extrinsic regulation, So this is regulatory mechanisms from external factors, so the autonomic nervous system and the endocrine system. And blood flow is adjusted to meet needs of the body instead of just for like a specific tissue in a specific region of the body. So here's how the sympathetic nervous system regulates the arterioles extrinsically. So that adrenal medulla releases epinephrine, and sympathetic postganglionic neurons release norepinephrine. So this is a GI or GQ coupled response. Norepinephrine and epinephrine bind to alpha adrenergic receptors on peripheral arterioles, which leads to vasoconstriction. On the other hand, epinephrine binds to beta adrenergic receptors, leading to vasodilation. So the two things I highlighted on this slide are the most important takeaways. Not the yellow, the like reddish and blue. It's important to understand that norepinephrine and epinephrine bind to alpha adrenergic receptors that leads to constriction. Alpha constriction, and then epinephrine binds to the beta, leading to vasodilation. So something to think about here is norepinephrine and epinephrine are neurotransmitters associated with the fight or flight response. So, like we talked about a few slides ago, during the fight or flight response, where do we think that we're going to constrict arterioles? Does anyone have any ideas? Yeah.
The GI tract.
Yeah, exactly. So remember, fight or flight, we don't need to focus on digestion. So we can constrict around the GI. Beta adrenergic receptor vasodilation in response to epinephrine would therefore dilate arterioles around like the brain, heart, lungs, skeletal muscle. Yeah, so that's just how like the body can regulate blood pressure through the sympathetic nervous system overall. And then at the bottom of the slide, there's a note that says there are more alpha adrenergic receptors than beta in the body, so it's easier to vasoconstrict rather than dilate, which means that it's easier to increase the total peripheral resistance in the arterioles in the body and increase blood pressure, or which is also known as MAP, or mean arterial pressure. So I took this from the videos when I took the course, and it kind of goes over what we just talked about. So you see the release of norepinephrine from the sympathetic postganglionic neurons, which causes vasoconstriction in alpha adrenergic neurons, and then epinephrine from the medulla, causing both dilation and constriction, depending on the tissue that it's like binding to the receptor in. And this is the main regulator of blood pressure in the body. Any questions on this so far? Or confusion? Okay. Next, we move on to capillaries. So now we're at the tissue. Capillaries are the site of nutrient and gas exchange. They are only the tunica interna. And they're a single cell layer thick. So there's no smooth muscle, which means that the body can't constrict or dilate them. They're just little passageways for one blood cell to fit through at once to allow for gas exchange between the tissue and the blood. So the red blood cells travel through a single file, which maximizes surface area for gas exchange. And then there are 3 types of capillaries based on the amount of tight junctions that they have. So first is continuous capillaries, and these ones have a lot of tight junctions. There's no breaks between cells. They're held together very tightly. And most tissues have continuous capillaries. So they're very sensitive about what they can exchange, because nothing can get in between the endothelial cells of the capillary to pass between tissues and blood. So anything that's leaving or entering these capillaries has to go through the cell membrane. And the cell membrane is very selective about what it lets in and out. So it's only permeable to things like gases in continuous capillaries. The second type of capillary is fenestrated. So these ones have an intermediate amount of tight junctions, so they allow for some paracellular movement. This just means that like some smaller substances can move in between the cells of the capillary or the endothelial cells in and out of the blood. So it's a little less selective. And some examples of things that can move between cells are small peptides or signaling molecules. And the third type of capillary are sinusoid capillaries. These have the least amount of tight junctions, so they allow for the exchange of large proteins in between the endothelial cells, and they have big gaps. And you see a lot of these in the liver. So here's just a visual: continuous, most tight junctions, fenestrated, intermediate, and then sinusoidal has the least amount of tight junctions. Okay, now we're going to get into exchange a little bit because that's what happens in the capillaries. So there are two types of exchange. You can do exchange by diffusion, which is the movement based on concentration gradients. So this is like more focusing on local movement. Diffusion is more on a smaller scale. And it's done by things that are membrane permeable, like gases. So diffusion is just like from a higher concentration down to a lower concentration, locally in a specific area in a specific tissue. Bulk flow, on the other hand, is more of a non-specific movement based on pressure. So if you have a high pressure gradient and you're pushing blood like through the arteries, then that can like push substances out of the vessels just because there's a high pressure, so they're kind of getting like squeezed out of the vessels. And this helps move things that are less permeable. So you can kind of think like bulk flow is moving things based off of force. Like the blood is like pushing so hard against the wall of the vessels that it's pushing a lot of materials out. When diffusion is just based off of a local concentration gradient of substance. Does that make sense? Okay. So now this is definitely an important distinction to have straight going forward. The direction of exchange is based on the type of vessel, or more so the side of the capillary that we're on. So filtration is on the arterial side of capillaries, and it's movement out of the blood into interstitial fluid. So it's really done on the arterial side. I should probably change that language. This is driven by hydrostatic pressure, which is the high outward pressure created by blood in the arterioles. So the blood is coming into the capillaries from a bigger radius, and then it's getting squeezed into a smaller capillary, so there's a lot of pressure. All that pressure is driving filtration, or substances to come out of the blood and into the interstitial fluid, which is just kind of like outside of the vessel. So blood is forced out, carrying some solutes into the interstitial fluid. Large proteins, like albumin we learned about last week, for example, stay in the blood. They cannot leave by filtration. And more fluid is filtered than absorbed. So now on the venous side of the capillary, we have absorption. And this is when we take some of that fluid that was filtered out and move it back into the capillary or vessel. So a way to think about this is like we're not losing it, we're absorbing it back into the circulatory system. It's on the venous side. It's driven instead by osmotic pressure. So if you remember when we did filtration, we left the proteins inside the blood. They could not leave and be filtered out. So those proteins or solutes in the blood are driving water back into the vessel osmotically. So you can think osmosis, like there's a higher concentration of solute in the blood. So the water is driven back in. Okay, so now we're into veins. So veins carry blood back to the heart from the tissues, and they also act as a conduit for blood flow. So they also store blood. Some information on the structure. They have a less developed tunica media, which means they have less smooth muscle, which means that they're not as well regulated by the nervous system. So they can't really constrict or dilate as easily as we saw in the arterioles. So that would mean that the arterioles are really the main regulator of blood pressure, whereas the veins just kind of are a passageway for the blood back to the heart. They have an oval-shaped lumen, which is more collapsed, and they have a higher compliance, so they're more stretchy, like a balloon. Because in some regions of the body, the veins really act as like just a storage reservoir for a lot of our blood. I think especially in the legs. And then on the bottom, I'm sure that you guys will look at this in the lab, and that's definitely something to know for the practical difference between artery and a vein. So the vein is just like the bigger, oh, no, that doesn't work. This like bigger space here, and then the artery would just be like this smaller circle.
What's on the next slide?
Yeah, there it is labeled. There you go. Yeah. And you can also see in the image on the left, like the thicker tunica media in the artery, which allows for better regulation, or the TPR, and then the smaller or thinner tunica media in the vein. Okay, so veins have two major functions. First one is just to act as a passageway to the heart for the blood. So they do this with their larger lumen, which tries to increase blood flow. And then they also have valves, which keeps blood moving in one direction. So this is especially important in the legs because their like blood flow is opposing gravity. So if we didn't have those valves, like blood flow would just like go back in the wrong direction, which would mess up the whole body's circulation. So they're really important to keep blood flowing back up towards the heart. And then varicose veins are when you have valves that are stiffened and damaged due to aging. So that causes veins to swell and widen because a lot of blood is like pooling there, like the valves aren't opening and closing correctly. A second major function of veins is that they act as the blood reservoir. So they hold 60% of the body's blood. And they can do this because they have high compliance. They can stretch and accommodate large volumes of blood with a relatively little increase in pressure. So we just talked about, or earlier we talked about arterial regulation. So this is kind of how blood is driven back to the heart from the veins. So there's they're only extrinsically regulated. They use the sympathetic autonomic nervous system, and they have alpha adrenergic receptors, which, when bound to, decreases their compliance. So that makes them a little bit less stretchy, which can help kind of push blood back up to the heart. But since they have a thinner tunica media, it's a lot less intense than the arterial regulation. Then they also have a skeletal muscle pump, which just means that like they're placed a lot of the times between skeletal muscles. So when the skeletal muscles contract, it helps push the blood back up to the heart. Third, another driver of blood back to the heart is the respiratory pump. So this just means that expanding the lungs in the chambers of the heart kind of creates a subatmospheric pressure that helps drive blood back to the heart. So creating like a negative pressure by increasing volume helps drive the blood back. If you can think like when the atria empty, there's something needs to fill that space. So that negative pressure helps drive the blood back up to fill that space in the heart. So this slide I did not completely finish, but there are two like major terms I'd say to be familiar with like in regard to the physics of blood flow. The difference between resistance and tension. So resistance is just opposition to flow caused by friction, whereas tension is like stress on the wall, or outward pressure created by the blood. So I did not finish this slide, and I can finish it. And if you want, you can also access this information in my notes too. So then here's some, or a layout of the 3 layers of blood vessels in general. The tunica interna is just endothelium, or like epithelial cells. So capillaries are just tunica interna. Then there's the tunica media, which is the largest and most developed layer. Like we've talked about, they're very well developed in arteries, and it's present in veins, but less developed. And this is smooth muscle and elastin fibers. And then the tunica externa is connective tissue. So this is like collagen that helps the structure of the vessel. And arteries have extra elastic proteins that aren't in veins, which may help decrease compliance. So next is compliance. This just measures how easily a vessel changes its size in response to changes in pressure. So as we've talked about, arteries have a low compliance due to their large tunica media and high presence of elastic fibers in the tunica externa. So you need a large change in pressure to maintain a change in shape. So they don't want to stretch out. If you get a lot of blood in the artery, they're going to want to recoil, which helps send the blood throughout the body and create like the circulation of blood. Veins, on the other hand, have high compliance. They can stretch, store a lot of blood, and aren't really contributing to blood pressure as much. So the 2 main functions of compliance are to distribute blood to the body and to act as a pressure reservoir. So allowing that recoil is what sends blood throughout the body. It acts as a secondary pumping action. So the heart pumps the blood to the vessels, but having a low compliance kind of helps continue to pump the blood through the body as like a secondary pump. And this is known as pulsatile blood flow in the arteries. So during diastole, the arterial wall recoils. So when the heart is relaxing, after the arteries have just received a lot of blood, the artery wall then recoils, acting as like a secondary pump to the heart. Okay, got some top hat questions. So I don't think, can you guys see these yet? I don't think. Okay, so I'm gonna try to assign them as homework so then I can see what you guys put for the answers. Okay, I think it works. Which is correct about vessels?
I'm still looking for it.
Okay. Yay, it worked.
I'm gonna try cooking your link instead. Oh wait, no. Oh.
You have 10. We only have yours. Okay. Does anyone? So there are kind of two parts to this question: the compliance and the lumen. So does anyone know if arteries or veins have a lower compliance? Yeah. Yeah. Right. So the way I kind of think about this is compliance is kind of like a synonym. I mean, it's not, but you can think about it in your head as like stretchy. So veins have a higher compliance. They're able to like stretch easier. And then the second part of this question is about the lumen. Do arteries or veins have a larger lumen? Does anyone know?
They have the smaller lumen because they're not stretching, like the veins are.
So which
the
one
veins
has?
have the larger lumen.
Yes.
Yeah. Because
Yes.
They stretch.
Yeah, the veins have a larger lumen. One, because they stretch, and two, because they have a smaller tunica media.
Right. That's it.
Yeah. So D was the correct answer. What tunica layer is less thick in veins and more thick in arteries? Okay, yeah, we've talked about that a lot. The tunica media is thicker in arteries and less thick in veins. Fenestrated capillaries. Okay, yeah. So fenestrated capillaries were the intermediate capillary with like some tight junctions, which allows for some paracellular movement or movement around the cells of small peptides and signaling molecules. Continuous capillaries are the ones that only allow for diffusion. I saw a couple of you clicking on that one a little bit. But if I asked about continuous capillaries, this correct answer would be B. And then C would be sinusoid, I think is what it is. Capillaries. Arterioles can alter TPR by So that's right. The arteries constrict or dilate based off of blood flow or the need of blood to certain tissues or kind of the whole blood pressure response, as we talked about, like intrinsic and extrinsic regulation and local vasoconstriction, dilation, and then like the fight or flight response. Filtration occurs on the blank side of circulation, while absorption occurs on the blank side of circulation. Yeah, so any form of like artery for filtration is correct. So if you remember, filtration is driven by that hydrostatic pressure of blood coming from the arteries into the capillaries. So things are being pushed out of the blood, whereas absorption is more on the venous side, where water is driven back into the blood by osmotic pressure. I spelled that wrong. Absorption of water into the blood. So again, filtration is first on the arterial side, pushing substances out, and then on the venous side, water is absorbed back in, driven by like the proteins that were left in the blood. That's creating like that concentration difference almost that pulls the water back in. Any questions on filtration or absorption? Okay, cool. Place the vessels in order from path of blood from the tissues to the heart. Okay, so starting at the tissues, that's supposed to say venules, not venues. Starting at the tissues, we have the capillaries that like are covering the tissues. These bring the gases for exchange. So that one is going to be first. And then from the capillaries, we merge into venules, which then merge into veins, arrive at the heart at the pulmonary vein. Some are at the heart, those are kind of synonymous. And then we go to the lungs, get some more oxygen, and are pumped out via the aorta into systemic circulation, where we first go to arteries, then arterioles, and then are back at the capillaries and start the cycle over. Any questions on that? Okay. I think we have 4 more. Arterioles regulate the radius size to help blood circulate through the circulatory system. In other words, the size of the arterial's radius determines, and it's inversely proportional to. So I didn't really go over the physics part. But yeah, it's in the notes. Cool, you guys got it. So radius is inversely proportional to resistance. Which is false about blood pressure.
Oh. I put the true. I put the one that was most true. I'm tired.
I am too.
But I knew it was true.
Yeah, those will always get you, like, which is false.
Yeah.
Nice. So there are kind of two ways to represent blood pressure that are good to be aware of. You have the MAP, which is the mean arterial pressure, which is the average. And this one's used more in clinical settings. And then the one that a lot of us are familiar with is the systolic volume over the diastolic volume. But neither of those are diastolic divided by systolic. So that's false. And then the other one is true. What is the purpose of extrinsic regulation in veins? What effect does this have on compliance? Perfect. So we want to decrease compliance or decrease stretchiness to help push blood back up to the heart. Last one. Besides extrinsic regulation, what two other mechanisms do veins use to maximize return? Okay, yeah, so I was going for skeletal muscle pump and respiratory pump. Valves are also pretty true because those help make sure that blood goes back in the right direction to the heart. One that I did see that I don't like is constriction and dilation, which is a good distinction to make because remember veins have less tunica media. They're less regulated by constriction and dilation. They can't really constrict or dilate. That's more in the arteries. Yeah, so that's everything for you guys. Let me know if you have any questions, and thanks for coming.